Greetings everyone, and today we are going to start cytoskeleton. And cytoskeleton, before discussing cytoskeleton, let us discuss the terminology. Cyto means cell, skeleton means the framework. So cytoskeleton is defined as a framework structural framework of the cell. As it is a structural framework of the cell, it is present within the cell. Its occurrence is within the cell. Now, it is the structural framework of cell, which is made up of network of fibrous proteins, which includes several types of fibrous proteins, including tubulin protein, which is present in the microtubule, which we are going to discuss here. Fibrous proteins. Now, these fibrous proteins are in different range. First, we're going to discuss about how the cytoskeleton work in the cell. Cytoskeleton are basically scattered all over the cell. These are responsible for a few things. First of all, cytoskeleton assists in shape maintenance, apart from which it provides and assists in the shape maintenance, as well as it allows motility in cell, motility in both cases, motility of organelles such as cilia, flagella, as well as for the streaming movement of cytoplasm. Cyclosis is also done by cytoskeleton, apart from which it also helps in cell wall deposition, apart from which it also helps in the process of cell division by the formation of spindle fibers during cell division, in which the spindle fibers are formed, which attract the centromer towards them, due to which centromers are attracted and then um, attracted to the opposite poles, and then the, the nuclear karyokinesis takes place by formation of a new nucleus with one half of the replicated chromosome. Apart from which, when it comes to its other function is holding. It anchors to the cell organelles. It anchors to the cell organelles, as you can see in the diagram. That's here in the diagram. There is the structure of cytoskeleton. Now, inside the structure of cytoskeleton, the mitochondria is anchored to the cell or anchored to the cytoskeleton. It is anchored to the cytoskeleton. Similarly, ribosomes are anchored, polysome is anchored, as well as these are the organelles. Endoplasmic reticulum is anchored within the cytoskeleton. Now, when it comes to the cytoskeleton, here we are having a few things more. That here cell division, it anchors to the cell organelles, apart from which it is present within the cell. That is why it is on the cytoskeleton. Apart from which it also acts as a mechanical support, but here we are going to majorly discuss about maintenance and mechanical support as well. On the other hand side, when it comes to its fragments, it is made up of three types of fibrous proteins, three network of fibrous proteins, which include the microtubules, microfilaments, as well as the intermediate filaments. Intermediate filaments. Now, first, let us discuss about microtubules in this video. So here, microtubules are basically made up of, these are the network of tubulin protein. These are the network of tubulin protein in such a manner that each microtubule contains around 100 to 1000 tubulin subunits. Contains 100 to 1000 tubulin subunits in such a structure that tubulin protein is made in such a structure that here one alpha tubulin protein, one alpha tubulin protein. Tubulin is basically a dimer of two subunits, which includes the alpha tubulin protein and beta tubulin protein. These alpha tubulin and beta tubulin protein forms a dimer which is known as the heterodimer of the alpha and beta tubulin, which causes to form a single tubulin subunit. Now, this single tubulin subunit is arranged in the structure of a polymer. It is arranged in the structure of a polymer by the process of polymerization. For suppose this is one tubulin subunit, there is another tubulin subunit, after which there is another tubulin subunit, and so on. There is a complete chain of polymers of tubulin subunits, and that complete chain of polymers of tubulin subunits is known as the protofilament. And within the protofilament, this protofilament is what? It is a parallel, it is a polymer of the tubulin subunits, which is arranged parallel, which is arranged parallel to it is arranged parallel to the which is arranged parallel to each other. These are these protofilaments are arranged parallel to each other in a group of 13. These are arranged parallel to each other in a group of 13, which collectively forms a structure which is known as the microtubule. The microtubules. So when the tubulin subunits collectively form a polymer of subunits, it forms a protofilament, and when 13 protofilaments are arranged parallel to each other, they cause to form when 13 parallel protofilaments are organized together, they cause to form a structure which is known as microtubules. Now microtubules are present in both animal and plant cells. When it comes to the diameter of microtubules, it's around 25 nanometer, or we can also call it as 250 angstrom. 250 angstrom. Apart from which, when it comes to its length is around 0.2 to 25 micrometer. Apart from which, when it comes to its structure, it is just like a hollow cylindrical structure. As you can see in the diagram as well, it is a hollow cylindrical structure in which it is formed in the shape of a cylinder, and in between the cylinder there is the empty space. This empty space is the hollow cylindrical structure which is present in between the microtubules. Apart from which, when it comes to another part that we are going to discuss here, is that here inside this structure we are having the um, several alpha beta tubulin proteins. As you can see in the diagram, there is alpha tubulin, beta tubulin, both form a heterodimer of the tubulin subunit polymerized to form the protofilament, and protofilament 13 protofilaments 13 parallel protofilaments are organized together in order to form a microtubule. Now apart from this, let's discuss some functions of microtubule as well. First of all, microtubule is a structure which helps in the movement of flagella and cilia in eukaryotes. Flagella and cilia in eukaryotes. So it helps in the movement of flagella and cilia in eukaryotes in such a manner that the flagella, cilia, basal body, centrioles are all made up of microtubules, due to which it helps in the movement of flagella and cilia. Flagella is commonly found in flagella is found in the eukaryotes in euglena, whereas cilia is found in paramecium. Both of these are having their structure made up of microtubules, which allows the motility movement of the flagella and cilia. Apart from which, when it comes to its other functions, microtubules, there are also microtubules in bacteria. I mean, they are not microtubules. In bacteria, there is flagella, but that flagella is made up of flagellin protein rather than tubulin protein. On the other hand side, a tubulin protein is having another function. The protein causes the tubulin protein causes causes the separation of separation of replicated chromosomes separation of replicated chromosomes at the time of cell division. At that time, it helps in the replication of chromosome at the time of cell division in such a manner that it helps in the replication of chromosome at the time of cell division. That here in plant cells, it is found freely dispersed within the cytoplasm, while in first of all in plant cells we are not having centrosome or centrioles. We are only having the structure of microtubules which is freely dispersed within the plant cell. On the other hand side, when it comes to animal cell, in animal cell it is found in the form of basal body, flagella, cilia, and in centrosome. So during cell division, the structure is in such a manner that here this is a cell. First of all, this is a cell during cell division. This is the chromosome. This is a replicated chromosome. Now, this replicated chromosome, as we previously studied, is attracted by some spindle fibers which are produced by the tubulin protein of the microtubule. Now, if it is an animal cell, in an animal cell, there is centriole. Centriole is having tubulin protein inside it. due to which the tubulin protein produces several spindle fibers, organizes the spindle fibers which are taken out from the centriole, due to which these tubulin fibers are then attached with a protein, a disc-like protein over the centromer of chromosome, which is known as the kinetochore. Kinetochore, where the spindle fibers are with attached through the centriole. After which, when it comes to the structure of kinetochore, it is basically functioning in order to attach the spindle fibers with the centromer. So that is the function of kinetochore. And apart from which, also have an example of kinetochore, that here these kinetochore attached with the spindle fiber. Now this is the case for animals. When it comes to plants, in plants there is no centriole, due to which the spindle fibers are taken out from the tubulin protein, but this time these are taken out from the microtubules which are freely dispersed within the cytoplasm, due to which when as these microtubules are freely dispersed within the cytoplasm, as the microtubules are dispersed freely within the cytoplasm, these microtubules are dispersed freely within the cytoplasm. Microtubules are dispersed freely from where the spindle fibers are attached and arise and then attached with the kinetochore. Now, in both of the cases, there's also an enzyme, there's also a chemical known as colchicine, colchicine, which is a chemical which inhibits the growth of spindle fibers, and that is derived from a plant known as calcicum, calcicum. Colchicine, which is derived from a plant known as calcicum autumnal. Calcicum autumnal, which belongs to the family Liliaceae. It is the uh, same family which is having the which is having onion as well as garlic. Which is having onion as well as garlic. So this is um, a brief intro about it. And apart from this, what is its functions we have discussed here precisely. This is a structure of microtubule, and these are the functions such as movement of flagella, cilia, vessel body, helps in cell division, cell wall deposition, as well as helps in motility, as well as it helps in the process of formation of spindle fibers. Apart from which, it is having its process of forming. After which, that's all about this video. Hopefully, we'll meet again.